This podcast was produced by OAR FM Dunedin with support from New Zealand On Air.
Welcome to the OI podcast, the Otago Institute podcast, in-depth conversations with experts from the worlds of arts, sciences and Mataranga Māori, brought to you by the Otago Institute, the Otago branch of the Royal Society for Arts and Sciences. Kia ora, I'm Dr. Barb, and today in the studio it is my immense pleasure to welcome and introduce Jennifer Bannister. Jennifer, welcome to the show today.
Thank you.
And it is so amazing to have you here, Jennifer. How long is it since we first met? It must have been 1995.
Yes.
In the botany department.
I came to New Zealand in 94.
Right, yes. Oh, my goodness. So yeah, that's a long time. And I came to Otago University in 95 as well.
Yes.
So there we go. Our whole careers in New Zealand are simultaneous. So you started off with a BSc Honours in Nottingham. In botany,
yes.
But that wasn't in 95, was it?
No, it was 1960.
1960, which is quite a different environment to be studying at university there.
Oh, completely. Yes.
And I imagine
there were only 2,000 students. And
at Nottingham University.
Wow,
now. Yeah.
2,500, I think.
Right, yeah. And there's stories about your honours class, of course, because that's where you met your eventual husband, isn't it?
That's right,
yeah.
Yes, Peter. There were 7 of us.
Wow.
And 4 of the 7 ended up in New Zealand, believe it or not.
Wow, I didn't realise that. I knew, of course, that you and Peter
and his first wife.
Right.
And then me and somebody called Mike Baxter, who was in the North Island. But I think Peter might have met him once. But
that's quite a hit rate, isn't it?
4 out of 7.
Out of 7, yeah. And quite a small class for the honours class there, I suppose.
Those were the days.
Yeah. And were you one of 2 females in that class, or was there more of an even split than that?
More of an even split. I think there were probably 3.
Yeah, because it's interesting with botany because it has been, ups and downs as all subjects have, but it has been one of the sciences that has been more open to female students of botany throughout the kind of time, right?
Yes.
I guess
from Victorian times.
From Victorian times, yeah. It was that whole collecting plants, I guess.
It was a suitable job for women.
A suitable job for women, yeah. And that whole, anyway, very strange. But today we're here now. You've got 3 topics, so we better not muck around too much.
No.
We've got 3 topics because you have had quite a varied career here in New Zealand. Of course, I met you in the botany department, and you're still in and out of the botany department, aren't you?
Not as much as I've got a little bit older.
Right. But and transferred a little bit to the geology department, might I?
Partly, but I have always done all the work I've done at home.
All right. Now, I've
set my own study up with microscopes and cameras.
Oh, right. Okay. Well, that's very sensible. There's not a lot of commuting in that. That's the way to go.
Well, I found when I used the microscope in botany, the research microscope, different people had been using it, and it was always set differently.
Oh, yeah, that slows you down,
I just bought my own instead.
Right, yeah. And it's true, I always associated you with small things.
Yes.
And the first of our topics today is lichens, and then we're going to talk a little bit about paleofossils, and of course the new book, and the excitement of the new book that's out. And then leaf-tooth hydathodes. Oh my goodness, how ridiculous do I sound? We'll get back into those in a later date. We'll start off with lichens because lichens are huge. As in, they're very abundant. There's so many of them.
Yes.
And I imagine elsewhere in the world as well as New Zealand, but possibly New Zealand in particular, for a long time they went very unnoticed, just written down as lichens.
Yes. For instance, in the United Kingdom, lichens have been worked on probably since 1700s, certainly 1800s, and but not in New Zealand.
No.
New Zealand's a good size for looking at lichens, really, because you can get around And one of the first things I did after learning, you have to learn a few lichens and how to identify them.
You do.
And I then decided that I would like to just specialise at that moment in one particular genus, the genus Ramalina.
Right.
Which had just been worked on by Dan Blanchon from Auckland as his MSc topic. So
right.
He'd worked out the species, and it was a nice group that was well worked out, and I decided to specialise on them in their distribution.
Right. So he was working on the taxonomic relations between the species within that group.
Well, describing all the species of Ramalina in New Zealand.
Yeah, interesting. And then you decided to take up the baton, as it was.
What I did here was have a look at some of the herbaria specimens of them. But the most exciting thing that I did with Peter, my husband, was to do the distribution of the genus throughout the whole of New Zealand.
Wow.
We used, weekends, long weekends, holidays, and we went on trips and covered the whole of, well, I say the whole of New Zealand, but,
I imagine
as much as we could.
Yeah.
And there was the first paper I was ever associated with here was the distribution, habitat and relation to climatic factors of the lichen genus Ramalina in New Zealand.
How many species in the genus?
I don't remember. I count them if you like. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15. 15, but I think there are a couple more that we've added to them.
Yeah, that always sort of happens, doesn't it? The more something's studied,
it's an easily done, if you like, group.
I mean, it's not hundreds of species. Yes. It's not like eucalypt where you've got 800 species that are all hybridising.
Yes.
I wonder if we could take just a step back there for those that are less familiar with lichens and just go through the kind of what makes a lichen a lichen.
A lichen is a symbiotic association of a fungus with an alga.
Yes, it's very strange in that it's sort of two separate organisms, but then they make the resulting organism has its own taxonomy because not all fungi associate with all algae.
No, the different genera of lichens tend to have their own particular alga that they associate with, and there are a few that are used mainly.
Right.
But that can vary. Sometimes it's a green alga, sometimes it's blue-green.
Wow.
And the fungus makes the body of the lichen.
Right.
And inside are the algae, which are associated with the fungal hyphae.
Right.
And the algae, of course, are photosynthesized.
So that's where it's getting its food from. And I guess the fungal part is...
Yes, the minerals.
It's getting the minerals.
Nitrogen and so on.
It's quite a clever construction, isn't it?
It's a very clever arrangement.
And it's interesting that, I mean, I guess similar to corals in a way.
Yes, or mycorrhizae, if you like to think.
Yeah.
In
plants.
For tree roots.
Yeah. That kind of two things that kind of are coming together. But you find them reliably in those pairs enough that you can identify them in those pairs all over the country, and they're very stable evolution. Do we know how long those pairs have been partnered up? Is there evidence from DNA? That's possibly completely outside of your expertise.
It is outside of, and the evolution is not of lichens is not well known from the past because it's very difficult from fossils to say what is a lichen because it's not well preserved.
Right. Yeah. Of course.
The fungal hyphae and the green algae, to show that it was a lichen, should be preserved together, and you don't really find that.
Yeah, no,
there have been cases of fossil lichens being described, but I'm never quite sure.
Yeah, and I guess also
good.
They're so small, aren't they? And so, and quite often, quite thin, fragile.
Yeah.
Being turned into a fossil if you're a robust Tricera.
Yeah, I mean, there's no hard bits.
Yeah, exactly. If you're a T-Rex thigh
Yes.
bone, it's much easier to be turned into a fossil or a skull, indeed, than it is if you're the tiniest little microscopic.
What can be done, of course, though, some of them are preserved in amber.
Oh, wow. I didn't realise that there were lichens in amber. You always think of Jurassic Park and you think of dragonflies and mosquitoes and things. I know Jurassic Park is absolute rubbish scientifically, but
you get pollen.
Yeah, of
and
course.
flowers.
Yeah.
But I mean, in amber, but not very many.
Yeah, I know that's interesting. Have you got any amber with lichens in them? No.
No.
Not an amber collector.
New Zealand does have amber.
Oh, right. Up with the kauri gum.
Yeah.
Forebears of the kauri gum.
But the kauri gum is present day.
Yeah.
The amber I'm talking about is preserved resin.
Right, yeah.
From conifers.
Yeah.
Not quite knowing, possibly Oricaria.
Yeah. I'm just assuming that they were the forebears of kauri.
Possibly agathis, yes.
Anyway, so your interest in lichens, you started off with this distribution of Ramalina. I wondered if you could describe
I can just show you the map that we did.
This is quite cool. Oh, so they're absolutely everywhere. But a lot of coastal ones there. Around this one here, this is your herbarium records and your sites visited. I guess lichens go all the way up to mountains though, don't they?
Oh, yes.
Yeah, they're sort of everywhere, aren't they? And particularly in stressed habitats, but then in full-grown forests as well, on trees and on rocks. And
yes, they need water. So
right, because the fungal...
very few of them are specialised for dry conditions.
Right.
So most of them need a certain amount of water, but also, of course, they need light for the algae.
Yeah.
So and they don't want too much water either. So they're
because they don't have a big root structure, of course, being that fungi. Just a little hyphae at the bottom there, so they just are collecting. How do you identify them? If you're looking at them, you were talking about your microscope there before.
It does depend. You've got three sorts, basically, of lichens. You've got fruticose ones, which are branched, sort of shrub-like,
right,
that growing on bark. And you've got foliose lichens, which are leaf-like, and they also grow on bark or on rock or on the soil. And then you have the crustose ones.
That's like that orange one that's splattered all over concrete, isn't it?
Yes.
And they just sort of look like someone spray painted graffiti on things, but really
and some of the
lichen.
alpine rocks have got sometimes called the paint lichen, a lechinora that is great white splotches of on these subalpine anyway rocks.
Yeah, and of course lichen is famous for being the food of reindeer.
Yes.
Well, not here, obviously. The northern hemisphere.
I haven't noticed any reindeer lately.
Yeah, no. But no, in the northern hemisphere, it's one that, because it's that whole thing with, when they had Chernobyl and they had all the nuclear kind of in the air, and the
came to the UK as well.
Oh, it did, of course, yeah. It went right across to the Welsh lamb.
That's right.
You
couldn't eat Welsh lamb.
You couldn't eat Welsh lamb for a bit there. And I know the reindeer, because they eat lichen, and the lichens were really taking up, because of the way they take moisture out of the air, I think it was, they were really taking up a lot of the radioactive and the thing. And amazingly...
well, of course, they have to take in carbon dioxide.
Of course, like all plants.
For photosynthesis, yes.
And of course, there's that amazing thing where it stopped around the entire boundary of France, because France doesn't believe in anything bad about nuclear. There was some strange papers at the time that had, quite a wave of radioactive atmosphere coming across. Anyway, I diverge. I do know that there's some chemistry involved sometimes in lichens. Have you dabbled in that?
Yes, you have. That's part of the way that you identify them. There are some chemicals we use and do spot tests, and that's it's actually quite useful for identification. But mainly,
sort of things go different colours.
TLC. Thin layer chromatography.
I thought it was tender loving care.
No, not quite.
Can you explain to me? Can sort of expand on what
Well, you extract part of the lichen in acetone.
Right.
And then you use chromatography paper and make spots with a capillary tube of the extract on the paper. And you try and get a concentrated spot of the extract. And then you put it through various solvents and under UV and so on.
And how it reacts to them.
And the spots move. Obviously, as the solvents rise, these spots separate out into their different chemistry.
Right. And then you use that to identify which species it is based on how the type specimen did the same things.
Yes.
Yeah. Interesting. But do you need to do that? Are there some lichens that are very easy to identify just from people just wandering around and going, that's...
The fruticose ones, the bigger ones, they tend to be able to identify from there. But when you come to the crustose lichens, they are more difficult. It's quite difficult to get good chemistry because the crustose are growing either on bark, and so you've got contamination from bark or contamination from rock.
Right.
But it's possible to do some. But this is the latest lichen group that I've been working on, Purchusaria, which to identify we've used mainly the spores.
Ah.
And
so you're looking at the
Purchusaria, the spores are quite large, but large comparatively.
Yeah.
The largest spore in Purchusaria is 300 microns.
Right. So you're looking at the morphology of those spores under the microscope and using that.
And the size.
And the size.
The size mainly.
Right.
The shape and the size. But the morphology, in Purchusaria, they're all simple spores.
Right.
But in other crustose, they may be septate.
Right.
Or they may be long and spiral-shaped. Or they may have double sets of cells.
Wow.
And so on. And with the Purchusaria, we've looked at the spores and the spore size.
Oh, wow.
And you can get them grouping out quite nicely by size.
Yeah, they are very different, aren't they? I'm just looking at this graph here, and you've got a very neat little diagonal line there, and coloured, so they are sort of round, aren't they? Roundish. No, that's 100 on that scale and 300 on that scale. So they're longer than they are wide.
Yes, they're ellipsoid.
Ellipsoid, yeah. But they are quite well distinguished. Like the colours there, obviously, the spores, the different species. So they are very well described just by the size of them.
But one of the other very interesting things which I found out is that the structure that produces the spores, the ascus, as in fungi, because the reproduction is fungal reproduction, and the spores form as a result of meiosis.
Right.
And most meiosis has a two-stage process. First stage where the chromosomes are halved, and the second stage where you double up. So the first stage of meiosis in these in the ascus would be to 4 spores from one original cell, 4, and then some of them don't go beyond that, but others go as far as 8, which would be the second division.
Ah, right.
And that is very useful because you've got species with up to 4 spores and species with 5 to 8 spores.
Crazy.
And so using those two things with the actual length and width.
And you can just identify the species by that. Are there ID books out now for lichens?
Oh, yes.
So there are...
The flora is still the main one.
Right, yeah.
And you would have to collect papers for individual genera and so on. This material isn't published yet.
Right, yeah. And it's amazing you're still publishing things, Jennifer. You're so dedicated to the cause. And I imagine that just the difficulty of identifying them makes them difficult to study, and so that makes it difficult to learn, about, and particularly in a place like New Zealand where the number of people available to study any taxon is smaller, and there are a large number of...
And there's not much money available.
Yeah, and there's a large number of species that haven't had that history of being studied for 300 years already. So it's, everything's quite new, and the chances, I imagine, of discovering new species
you discover new species and new distributions. And
so that's always exciting, isn't it? Have you got any named after you?
No. Nobody's done that.
Oh, no. Well, see, that's the trouble, is because you're the one working on them, and people don't tend to name them after themselves, do they?
By the way, I just want to show you that as a photograph.
Oh, wow, that's pretty cool.
One Purchase area ascus.
Right.
With 2 spores.
Yeah, and you can really see
So that will be one of the group that only have the first meiotic division.
Wow.
And not the second.
Yeah, no, you can really see it very clearly there. And these are from your microscope when you've just taken an image of them, are they? Yeah.
Yes.
It's very cool. Yeah.
I'm even trying to use a bit of AI at the moment to try and improve some of them, because
Jennifer.
Slides from slide preparations can be very mucky.
A bit kind of blurry and stuff. That's amazing.
And so with AI, you can clear the background, put another colour background on, but leave that. That has been done a bit like that.
Right. Oh, that's very clever. And is there special AI software that does that, or just use... Wow.
Photoshop, Lightroom.
Wow.
And others.
And others. Oh, that's amazing. I'm very impressed there, Jennifer. I don't want to comment on other people's ages, but I suspect that you're slightly older than my parents, who have trouble...
I'll be 86 next week.
Yeah. And obviously have no trouble with cell phones, texting, and all the way up to AI. Anyway, hey, I'm going to move on, unless you've got final thoughts on lichens, I'm going to move on to the paleofossils, because, of course, you've been involved in this Foulden Maar, which is amazing. And there is this Fossil Treasures of Foulden Maar, this beautiful book, which Daphne Lee and... How do you say it's Uwe, isn't it?
Uwe Kalfus.
Kalfus
is German. He's done a lot of the
little insects and things.
Amber as well as insects.
And John Conran
from South Australia.
Yeah, and then of course you've got, this is a beautiful book, this, and of course it's very dear to our hearts here in Dunedin and Otago, because Foulden Maar is right here. And of course the big, public outcry when it appeared that it might get mined up for ridiculous purposes. I can't even remember what they're going to use it.
Most of the photographs in there of the leaves, and particularly of the cuticles, are my photographs.
Wow. The leaves are so amazing. I've just turned to the leaves here, folks. Now, what I want to ask you about the leaves is, in the photographs, they are different colours. Is that real, or is that put on after in the Photoshop? Like this one's bright yellow with darker veins.
Those leaves are as they are preserved but cleared by chemicals.
Right. And the material that you're looking at here, is this actual leaf material?
The cuticle, upper cuticle and lower cuticle, not the cells, it is just the cuticle that is preserved, but the cuticle is a map of the cells that are present.
It's amazing, isn't it? And, so the cuticle is just the layer that's on the outside edge of the leaf, and it's made of slightly sturdier stuff than the inside, because cutin, because its main function, I imagine, is protecting the leaf.
Yes, it's a waterproof layer, because
it's a bit like our skin.
You only had cellulose, if you think of things like mosses, they can only live in very damp places.
That's right.
Because they'd lose too much water elsewhere. And leaves need, obviously, to take in carbon dioxide and lose oxygen and water vapour.
So they need...
So they have holes in the cutin, stomata.
Stomata, yeah. And just the shape, like you've just got the edges of these leaves and all the veins and where you've blown them up. But you can see absolutely clearly, like this is a fern,
Yes.
which looks absolutely like you've got here Davillia-like sterile frond, and there is a picture of Davillia tasmanii next to it, which looks as though it's the same species.
Yes. These are about 23 million years old.
23 million years. It's incomprehensible that amount of time ago, I think, to myself, and I think to most people, because it's just got so many zeros that it just goes into a big box that I call long time ago.
Yes.
But if we think 23 million years ago, could you say a little bit about how New Zealand might have looked? Because 23 million years ago, that's back to the time of the dinosaurs. No. Dinosaurs stopped at 66 million years ago.
Yeah, Cretaceous, they stopped.
Yeah.
So mainly the Jurassic.
Yeah, so this is sort of a third of the way back to the dinosaurs that we think of as, the big dinosaurs. But still a very long time ago.
Yes.
And
one of the things that I enjoyed doing on the Foulden thing, apart from the fact that I did all the work on the leaves, of clearing the leaves, clearing the cuticle, mounting them, and then photographing them so that they could be identified. But one of the things that I really enjoyed doing was finding out that Foulden Maar in those days was a Laraceae forest.
Wow.
Because we had about 10 species of Laraceae and all the leaves. Now, if we'd left it to the palynologists, they would have
pollen
said,
people.
They would have said it was a podocarp notophagus forest.
Ah, interesting. Just because pollen is...
podocarp and notophagus pollen is distributed in the air and can travel for, say, 25 kilometres. So there, in fact, were no notophagus leaves at Foulden Maar and very few podocarp leaves.
That's interesting.
So
it's
it
one
was
of
the
big
leaves, you see, that told you what was there.
Yeah, because they're more likely to have dropped from the tree that was right there.
Yes.
Onto the ground and then into the Foulden Maar.
The point about Laraceae is that their pollen isn't well preserved.
Right.
So we wouldn't have found that out.
That's very interesting, isn't it? And I guess that's one of the special things about Foulden Maar, that those very thin layers that are there at Foulden Maar allow things as delicate as these leaves. Because I'm amazed here, just looking through this thing here, there's over 800 leaves that you've got recovered here, it said somewhere. 800 individual leaves and cuticles. That's in this photo. There's a photo of you here with your microscope.
Several photographs in there.
I know, it's amazing. But I was just thinking that is quite amazing, isn't it? These ferns just amaze me. You can see all the little sporangia on the backs of them. It's absolutely
and the spores. You can extract the spores, and also
amazing.
It wasn't just leaves. We did find flowers,
which is very unusual. I mean, how often do people?
They do.
Do people find flowers?
No, they do. They do.
Really?
In certain circumstances, flowers are quite common.
What are those circumstances?
Not in New Zealand particularly, and often they're not able to be identified. But this was our first flower, the first flower in New Zealand found with stamens and pollen grains.
Wow, the morphology and paleoenvironmental context of Fouldinia staminosa, a fossil flower with associated pollen from the early Miocene of Otago, New Zealand. I might put a link to that on the episode description. But obviously that whole genus is named after Foulden Maar.
Yes.
And I just noticed here that there's a fossil flower and pollen grains of fossil Luzuriaga peterbannisteri.
Yes.
Which must have been named after...
It was named after Peter.
After Peter?
Yes.
So that's very... Did you have some hand in that, or was that...
I think Daphne
Lee
and John
maybe
Conran suggested it.
Yeah. Oh, it's lovely. It's a beautiful little flower as well. Well, the flower that's next to it that's like what it would have been.
Yes.
The fossil itself is...
Well, flowers are quite delicate,
They
Barbara.
are very... I'm amazed that there's any fossils of flowers. I could just find that incomprehensible, that something as fragile as a flower with the petals and the, the stamens and everything of that flower morphology that's so delicate. I mean, it's hard to get a flower in a bunch from a florist home and in a vase intact, I find, even if you're just going from the garden. But, these are tiny little layers of petal that have managed to be preserved and stayed there for 23 million years. I just find that incomprehensible. Mm. I think much more so than fish bones. Fish bones, they are fish bones. I'm amazed. Teeth. Yeah, and fish teeth. There, I'm amazed when they're all intact and they're all in the right place and they look like fish. But these leaves, and they just look like plants that are here now, and just the range of them as well. Like at Foulden Maar, there you've got.
I think if you turn back, the other way, there on the top of that one. What have you got?
Several leaves from the Foulden of the extinct species Logeriaga, Peter Bannisteri. The species name. No?
I thought it was another one because we did find on one particular leaf scale insects.
Oh, wow. Oh, that's craziest. Wow.
At the launch of the fossil book, the electronic one, that was the only planty sort of thing that was there, and that was my photograph.
Oh, wow. Because that's a fantastic book as well. I was at the launch the other day. That's a collaboration within the geology department there, but also with this 3D work behind it. So the whole book is just an e-book, which is fantastic. So it's absolutely open access. Anyone can go online. And I think if you just search geology department fossil ebook, you'll come up to it. And what's really cool there, and I hope that they're going to do some of the plants and the insects and the scale insects
the
as these 3D models.
Would probably work, but they're very, very small,
Yes. But, that's the thing about 3D modelling. Once you've modelled it, if you can get the resolution of the scanning, then you can blow it up. And that's one of the amazing things, because then you can sort of see it, 100, 1,000 times as big as it was. So you can see all the bits of it.
Yeah.
For showing kids, you obviously look down a microscope and you can see all the bits perfectly clearly. But I think it's amazing to have that resource there where people can get an idea of the real things and when you've got that life-size scale there as well. So some of those teeth, obviously not plant teeth, but some of those teeth are amazingly big. But I just think it gives you something that you can touch, because of course you can't usually touch fossils because they're either delicate or special or they're behind a box somewhere. And, these leaves, I imagine these are quite delicate still, are they?
They've probably mostly disappeared by now. They don't last, the Foulden leaves. It's not a lot of good collecting leaves from Foulden if you're just interested in the whole leaf, because they deteriorate very, very quickly.
Oh, so as soon as they're sort of out of the maar.
And in the air.
Oh, really? And then the air just disintegrates them. Oh, that's quite sad, isn't it? Mm. But then I guess that you're there. That's why you have to document them at the time. So
that's why the photographs are right from the start. It's very, very important.
And you do all the cleaning on site, do you? When you've done it, do you dig you take them out of the ground when you find them? I don't know. How do you find them? Do you find them in layers, just with lots and lots of leaves all together?
Yes, to get anything out of Foulden, you would have to take a digger in to extract some of the diatomite.
Oh, which seems weird, doesn't it? Such fragile
things.
And
then you can split up the diatomite.
Right. So the diatomite is stone that's quite solid, is it? Is it mudstone?
It's made up of what dropped to the bottom of a lake.
Right. Yeah, of course, it's the sedimentary.
The white layers are the diatom,
Right.
So that is a fossil in itself.
Right. And they give you the...
And then in the winter you get dead material, so
Right.
you get the black layers.
And then every year you can basically count the years
Yes.
because you get the seasonal
Yes.
white and black layers.
Yeah.
And then you very carefully prise the layers apart to see the leaves.
Yes, and all that preparatory work on the leaves would be done at home.
Right. So you just take
You can't do it. They're too delicate
Right.
for that.
So you take them home and then you prepare them. Oh, I didn't realise that once they had the air on them, they were disintegrating still.
They dry up and
Because they're so thin,
and
I guess. Yeah.
yeah, just break
Interesting.
up.
But there's quite a lot of the mass, like proportion-wise, still intact underground, isn't there?
Yes,
Yeah.
if we were allowed access.
Yeah. No, it's a bit strange, isn't it, really, the access thing? But this is amazing. And just the amount of information that you are able to get from these
Yes,
is
yes.
absolutely
Yeah.
phenomenal. And, that's the...
And since I did the work on leaf-tooth hydathodes,
Yeah.
I can now look back at some of the fossil leaves.
Right.
And one of the interesting things is there that the toothed leaves, the proportion of toothed leaves, say, in a forest, can indicate certain climate.
Oh, right. I'm going to go back to you there and I'm going to say, explain to us exactly what a leaf-tooth hydathode is. Hydathode. I keep putting an extra O in there. Hydathode.
And an R as well
I
sometimes.
know, it's ridiculous. I want to say hydro, but it's not. It's hydathode.
Hydathode.
Hydathode. Yes, explain to us exactly what they are, Jennifer, if you would.
It's better if you had some leaves here that were toothed.
Oh, right. I'll imagine I've got a leaf
With teeth.
with teeth, like
So
a wineberry leaf. Let's imagine a
Yes.
wineberry, because that's got the nice serrated
That's right, yes.
leaf on it.
Yes. So leaves are either entire margined
Right.
or they have teeth, which can be called serrations, but there's different ways of describing them. At the moment, I'm really happy just with leaf teeth
Right.
or leaf tooth.
Yeah.
Well, what we found, well, if I start out, this was Bill Lee,
Yeah.
and he's working with somebody in America, Tamo Reichgeld,
Oh, yeah.
who was here.
Yep. Yeah, no,
Do you remember him?
no, Tamo, yeah.
Yes. Well, they do climate things, you
Right.
see, and they got information from something or other, and the only thing they could link it to was whether the leaves had teeth, or the proportion of leaves with teeth or not.
Right. So you're looking at a
And so
whole
he
community
wondered
of
what was
trees and plants.
So I looked at teeth. Well, teeth without leaves didn't have them because obviously
Right.
it's a leaf-tooth hydathode.
Right.
And I have some pictures here. There is a magnified photograph with two teeth.
Oh, right. So they are quite prominent, and they've got this extra vein that comes in here, and they're much bigger than the hairs that you can see along the side of them, on the margin there, and very... Do you know what? Is there something solid in there in the tooth?
Yes, it's leaf tissue.
It's leaf tissue.
The same leaf tissue.
But it does give a sort of a solider bit there. Oh, I'm going to get Dommy to take some pictures of these and put them up on the thing. Leaf tooth with clear epidermis and the vein splaying out. I see. That vein does look like it's splaying out and gets wider as it gets
No,
towards the
that's better. The photographs, as printed, aren't too good, but you can see the splaying out there.
Yeah, gosh, it's amazing how well you can take these tiny, tiny little pictures, little things, and blow them up into these big pictures. So these little bits of this thickened tooth, that's the hydathode.
I like to think really of the leaf tooth as a hydathode, and
Right.
call them a leaf tooth hydathode.
Right.
Because
Where does
that-
hydathode come from? The etymology of the word hydathode.
Hyda is water from the Greek.
Yeah, and the thode?
Don't know.
Oh, sorry.
but it's water.
Right.
Water pore, because the ordinary lamina of leaves has stomata,
Right.
but it also has some things that are called hydathodes.
Right.
That's a stoma.
Yeah. With
But
the little
they
holes
also have
that the
some
water in
that
the...
look a bit like that, but are probably bigger and separated in a separate area. And they are probably for water loss again.
Right.
But this hasn't really been investigated.
Right.
Before.
So is the function of these hydathodes, is that for the water to run off the leaf?
No,
What's it for?
that's a drip tip.
Oh, that's a drip tip. What's the function of these?
To lose water.
To lose water
But this,
from the leaf.
This work needs to be done.
Right. Oh, it's interesting.
So it
eh?
hasn't. It has. I know what they all look like.
Right.
And what they do. But
So these you would expect to find them then in wetter areas where the plant needs to lose water?
Yes. For instance, the climbing brachyglotis,
Oh, yeah, yeah.
which grows in forests from the forest floor and grows up the trees. Well, the lower leaves certainly have these, because these leaf teeth, of course, really stay once they've been made.
Right,
But
yeah,
they
of course.
may be more important in the young stage, or not, maybe throughout the whole life. So all this is something that
Wow.
has not been really looked at. But you see, in a damp forest and the young plant, you've got a lot of pressure from the roots forcing the water up the seedling, and it
yeah, of
could
course.
be that the leaves could get waterlogged.
Gosh, isn't that weird?
No, it's quite sensible, really.
Yeah, I suppose it is,
But-
yeah. No.
A seedling doesn't have as far to go if it's forcing water up, so it's
Yeah.
more likely to be waterlogged than a tree of
All the way up, because,
20 feet or something.
Yeah, because then the problem becomes, can you get the water all the way up there? But you've sort of got this, of course the mature tree is related to the seedling, so they have the same engineering to some degree. I mean, they change as they grow older, but still within a certain limit. So it's really, I've honestly, I've never thought about the little teeth on the side of leaves and why some leaves have teeth and some have an entire margin, and whether there's any function
what
to it.
I've found is that most teeth, I think we found for the forest trees, was something like 96%. If they had, I reckon if they have teeth, they have leaf tooth hydathodes, but they may not be working in the adult form. And what you get, and I'm afraid you can't see very well, but there are water pores
On the tips there.
on the surface of the leaf tooth.
Right. Oh, that's very interesting, isn't it? And it does
And
seem...
that one is one water pore stained.
Oh, right. Gosh, and of course these are things that are more likely to stay intact in the plant fossils, in the leaf fossils, than other parts perhaps of the leaf. So they would give you some sort of... If they are related...
Because the leaf tooth is covered like the rest of the leaf with the cuticle.
Yeah.
So we have it, and we have the water pores. There's
And you can see
another one there.
Wow. So that is not a fossil leaf, though, is it?
No.
No, that's a real, like a recent leaf.
But if you look at any of the fossils in the book,
Yeah. Gosh, that is quite
that have-
amazing, isn't it? And so then you can get, because of course a lot of this work with the paleo fossils is about trying to understand the environment that was there at the time, and how that environment's changed and how that relates to the other things that are found in the fossil record there and the surrounding area. And of course, really amazing that work with the Laurelaceae, and that must be quite a common artefact of work that's based solely on pollen records, is that you can only talk about what you can find. Mm-hmm. And if a big group of species have pollen that just doesn't tend to fossilise well, then you're unlikely to find it, even if they are there. Yeah. So it's really amazing to have somewhere where you've got people studying the pollen at the same time as you're able to study the leaf fossils and put those two records side by side and match them up.
One of the problems is that the flora in those days are not the same as the flora today.
Of course, yes, because 23 million times
the
is...
pollen isn't the same, and
yes.
it's very difficult. We couldn't, in a lot of cases, identify, for instance, from the flowers, the flowers we found from the pollen.
Right.
Because of not being able to know what the pollen
was. It's funny, isn't it? You have to have a reference collection of some sort.
There is.
To refer it back. If something's not...
There's an atlas, but a lot of pollen, for instance, each pollen is named, but there's one name that keeps coming up, and it's just a generalised pollen for a group.
Right, yeah. No, which must happen all the time because, you just...
Yeah.
How would you know unless you know? And if you don't know, you can't.
And, of course, nowadays there's not much pollen work going on because we stopped drilling for oil, I think, haven't we?
Do you think that's what pushed the whole pollen expansion?
One of the pollen things, diatoms and forams as well.
Oh, right. Gosh, you've got to think, so there's always, it's funny, isn't it? Because, by and large, we don't think of the good things that the oil drilling gives us.
With the pollen and looking at the pollen you found in the layers and you identified the same pollen, you knew that was perhaps the same date and so on.
Yeah.
So it was a way of dating rocks as well.
Yeah, they sort of did these, what they call wiggle diagrams.
The different types of pollen, but they weren't interested in the history or the family relationship of these pollen grains that they'd got.
Oh, really?
So you need taxonomic palynologists.
And you can't do DNA from this fossil pollen, can you?
No.
Because it just isn't there. And same with these leaves. There's just nothing to do the DNA from, which seems strange because everything is about DNA these days. But it's interesting because there must still be, the things that we have now across the whole world evolved from something. And I guess some of those lines, looking like going the other way, some of those lines may have gone extinct. Yes. And definitely locally extinct. So they're just those species are no longer here in New Zealand because in 23 million years the climate in New Zealand has changed so much that the kind of descendants of those plants, we'll just talk about plants for now, have gone extinct, but they may still have descendants in other parts of the world. So I guess as you get a more complete, as people keep researching and keep finding more fossils, you might be able to fill in some of the gaps and kind of make...
There probably won't be much more pollen work done because of the lack of money in it. There aren't many palynology courses being taught in universities. Like there aren't any paleontology courses being taught in Otago at the moment because there are no paleontologists in the department. Yeah.
And you didn't actually study paleontology as an undergrad?
I was always interested in fossils, and I did take courses when I was in London, working in London. Right. There was Birkbeck College.
Right.
It was a brilliant idea. It was for working people who had a job, and they went in the evenings and on Saturdays to do work. And I did basic paleontology and the one-up from that there.
So sort of a lifelong love affair.
And I did a micropaleontology course at King's College, London. Yeah. And some courses that were run at the Natural History Museum in London on plant fossils.
Yeah. And do they have lots
And in fact, at university, in those days, we actually had paleobotany course.
As part of your botany,
as part of your botany degree. Yes, as part of the botany.
Yeah, because it's interesting that the paleo fossils, when there was a paleontologist in the geology department, that was the geology department, not the botany department. So even when you're looking at, it just depends how you split things up, doesn't it? Whether you're put all the plant things together or put all the technique things together. But yeah, no, it's amazing. There's so much information here. Have you studied? These are all from Foulden Maar. These leaves. Have you studied leaves from other places in New Zealand? Where else have you studied leaves from?
I'm afraid one of the problems of old age is memory of names and places.
Other places is enough. Sorry, Jennifer, I didn't mean to put you on the spot.
That's okay. There was a swamp down in Southland area that was a very interesting mine, but also Pico Pico, again, Southland.
Oh, right.
And that was very interesting because although the leaves were very difficult to extract,
right,
because they just weren't right, and when you did try and get some off, you only had one surface of the cuticle, and you need both surfaces for identification. But what I found there, and this was lovely, it's serendipity sometimes. The paper I wrote was Life on the Phyloplane, Eocene Epiphyllous Fungi from Pico Pico Fossil Forest, Southland, New Zealand. But although I couldn't extract the leaves for cuticle information, on the leaf, on the cuticle, there were leaf fungi growing.
Wow.
And these leaves were covered with them.
Isn't that
because
amazing?
It was very hot in those days in the Eocene, 35 million years ago.
Right, so it's even further back.
And wet.
It was hot and wet, so it's very jungle-like in Southland.
Hot and wet.
Imagine that, jungle-like in Southland.
For Pico Pico, you would have to go from the pollen record of what plants were found.
Right.
Taking into account what you know, but the leaves were covered
in these fungi.
And that gives you a very good idea of climate.
Wow.
Because some of those fungi you can find living on leaves in Dunedin at the moment.
It is amazing how much detail there is in them. It's quite crazy. Do you find does it take a long time to make one of these slides like this? Like, how long would it take to make that picture there?
I do it in stages usually.
Right. Right, because you're doing sort of the cleaning stage.
Yes, and then it might need to be cleared more in another chemical, and then washed and mounted and stained.
Right, so
then mounted. So it's done in stages.
Right, it's quite a craft, though, eh? I wonder if it's. I mean, obviously it's science, but it's sort of also a bit art, isn't it? Like just how you get the photo and what edge you get on it, and how they look, because they all, because you've got to show what bits are there, and you've got to show that the size and the shape of them, and they've all got their scale bars and the obvious the drawings that you've got next to them, which are, without reading the legend, I imagine you've drawn the basic features out of one of these slides. But just the preparation of the slides is quite a technical and quite an art type sphere. I just think it's one of the few areas where you're sort of still in that Leonardo Renaissance type mingling of art and science in equal parts in a world. I wonder if that's where kind of the lack of love in universities for it is partly responsible for it, because people get streamlined into a science route or an arts route, and I often think that we do a huge disservice to society and all the individuals in the society when we do that, because I think that I did a science degree, science honours and a science PhD, but I also did some arts papers, and I really appreciate the arts papers that I did do, and I think that actually I would have been a better scientist. Well, I was a better scientist for having done those, but I would have been a better scientist for having done more art subjects, and I really think that we should kind of go down that road where you have all arts students doing some science papers and all science students doing some arts papers. What's your thoughts on that, Jennifer?
Would never happen.
Would never happen. Oh, just imagine an ideal world where
Would never happen because it's all about money these days. It's all about getting a job and getting the best qualifications for a job.
Well, I don't know. It's not all about that. I mean, people want us to think that it's all about that. But, there's intrinsic societal factors and just knowing stuff. Because, how much of the work that you've done has been paid work that you've done for a vocation, for a job, and how much of it have you done because you just love doing it?
What I've done here, I've not been paid at all.
Yeah. See, this is.
I've not been paid, but obviously the chemicals and stains and materials were provided. But.
Yeah. But this is science for the love of science, isn't it? It's the most pure form of science there. Anyway, hey, it's been absolutely amazing talking to you today, Jennifer. And I'm glad that we cleared up what these hydathodes are. Oh, my gosh. But it was lovely talking about lichens because, of course, I think the first time I met you, it was over some herbarium specimens of lichens that are in the botany herbarium, and they were in tiny little tobacco tins.
Tobacco tins, yes.
And it was
the J.S. Thompson collection.
The J.S. Thompson collection. And it was this huge collection from a long time ago. When was he?
The 30s and 40s.
Yes, one of the very early collectors, and he had... because lichens are hard to put onto herbarium sheets, of course. That's one of the big things, because they're kind of crusty and kind of dry
you
sometimes.
You don't put them on sheets, you put them in packets.
And you put them in these little packets.
Yes.
And it's just difficult to make a collection of them because it's difficult to conserve them in, plants you can press and put them on the nice herbarium sheets and things. But he had them all in these little tobacco tins. And hundreds and hundreds I can't remember how many were in the collection, but I had a summer...
And who took them out of the tins and put them in the packets?
Yeah, me. I had a summer job. Oh, really? I had a summer job doing it and typing the night. No, I think that's how I met.
Yes.
I think that's how I met. We were in there. And,
yes.
And I hate to admit this, because very young at the time, the Gravy Mountains... He spent a lot of time tramping around what are absolutely not called the Gravy Mountains.
Garvey.
The Garvey Mountains. And I transcribed it wrong, and I think, anyway, and the producer's laughing at me. Slightly dyslexic there. I'd written out a whole heap of these labels, and luckily you caught that. Anyway, from the Garvey to the Gravy Mountains, it has been lovely speaking to you, and it's been it's 30 years, Jennifer, since we started with those little tiny tobacco tins in that little tiny room in the botany department herbarium there with that J.S. Thompson collection. And I imagine that you've used parts at least of that collection in your distribution data paper. No, because there weren't any there weren't any of the right species there. Goodness me. Anyway, hey, thank you so much for coming along today, Jennifer. It's been a pleasure. And I will leave us there. You've been listening to the OI podcast with Dr. Barb. If you enjoyed this episode with Jennifer Bannister, you will enjoy some of the other episodes. I'm sure we had a really great episode last year with Emeritus Professor Alan Mark, and we had the Christmas special with Janice Lord, and that was fantastic as well. And we had the Research Award winner Peter Heenan on, and he's coming along in August for an In Real Life seminar. So that will be fantastic. Thank you very much. Ka kite.
The shout out for the arts, sciences and Mataranga Māori.
You can support this podcast by becoming a member of the OI. Check out the Otago Institute for Arts and Sciences on Facebook or our website.
If you'd like to listen again to this episode or others from the series, look for the OI podcast on ORFM's website, oar.org.nz.
Brought to you by the Otago Institute for Arts and Sciences, the Otago branch of the Royal Society of New Zealand.
This podcast was produced by ORFM Dunedin with support from New Zealand On the Air.